right. Hi, everyone. Thank you very much to the organizers for inviting me to give a talk today. My name is Weighill, and today I'll be talking about some work I did in collaboration with both UNC Lineberger Comprehensive Cancer Center, where I'm a postdoc right now, and the Harvard T.H. Chan School of Public Health, where I was a postdoc with John Quackenbush previously. And so today I'm going to talk about gene regulatory networks and some modifications we've done to some existing methods to make these methods able to predict the regulatory relationships on an individual specific level and how they are altered based on the mutations that an individual harbors within their genome. So this talk will be divided into two main parts. The first, I'm going to discuss gene regulatory networks and how we've used them to look into differences in between the two main pancreatic cancer subtypes, basal-like and classical, and then also how we've tried to extend this method to incorporate the effect of genetic variants on these gene regulatory networks. So a valid question is, why should we investigate genome-wide gene regulatory relationships? And if one looks at the literature on complex phenotypes, we know that complex phenotypes tend to have their genetic underpinnings enriched in regulatory regions of the genome and not just coding regions of the genome. A simple example for this is in the phenotype for human height. If you look at GWAS signals for height, they are enriched in gene regulatory regions. Also, in the NHGRI GWAS catalog, more than 88% of single nucleotide polymorphisms are intergenic or intronic, so not necessarily within coding regions. We also know that complex phenotypes are associated with parts of the genome which span the entire range of the genome across chromosomes. So if you were to divide the genome into 1 megabase genomic regions and you looked at a phenotype such as schizophrenia risk, you would see that more than 71% of 1 megabase genomic regions have at least one variant which influences schizophrenia risk. And it's also known that transcription factor binding annotations can explain a substantial portion of disease heritability. So it is very important for us to consider not only the coding regions of the genome, the gene regions which are expressed as mRNA and then translated into proteins, but also to look at the regulatory regions, the promoters and their enhancers, which do not necessarily code for genes, but are nevertheless very important in controlling gene expression and play an important role in disease heritability and phenotype. Now, why do we want to look at gene regulatory networks? Is because these types of models capture the effects of these non-coding regulatory regions. And why relationships? Why not just look at gene expression as opposed to relationships between regulatory properties of genes? And this can be explored with an interesting toy example. So if we consider this example data here, where we have 9 individuals who are healthy and 9 individuals who have theoretically a disease, and we have here models of 4 different genes, and these heat maps show the relative gene expression of each of these genes across all of these individuals. And if we were to look at box plots of these genes, we see that none of them are differentially expressed between healthy individuals and individuals with the disease. So there's no differential expression. There's no signal showing us which genes are important in the phenotype of this disease when looking purely at their expression. However, if we look at patterns of the relationships between these genes, so particularly if we look at the correlation between the expression of genes 2 at a time, we can see that these are very, very different patterns in healthy individuals versus individuals with this hypothetical disease. So while there is no differential expression, there is clear differential co-expression. The relationships between these genes have changed. So it's important to be able to look at not only the expression of genes, but how the relationships between genes and the relationships between genes and their regulatory elements change. And we would like to use networks as ways to represent these gene regulatory relationships. And this is not a simple one-to-one relationship. You can have a transcription factor which regulates a gene by binding the promoter region of that gene and affecting the expression of that gene. However, some transcription factors also require a partner, a second transcription factor, in order to be able to together bind the promoter region of a gene and regulate expression. So these many-to-many relationships can be represented as a network. We represent this as one set of nodes of transcription factors and another set of nodes of genes, and we connect a transcription factor node to a gene node if that transcription factor will bind the promoter region of and regulate the expression of its connected gene. And we can look for these footprints of potential transcription factor binding sites in the sequence of the genome, and they're called transcription factor motifs. And there are pieces of software, there are hidden Markov models that you can use to try to identify these patterns, these footprints of transcription factors binding sites in the genome. Now, looking just at the footprints, the potential binding sites of transcription factors is not enough to call that a regulatory relationship, because, as we said before, sometimes transcription factors can bind on their own. Sometimes they require a partner to bind. And these patterns will also change depending on the cellular context, what tissue you're in, what state the cell is in. So we want to be able to incorporate other evidence to make this a more reliable model of gene regulation. So to this, we look to both protein-protein interaction and gene co-expression data. So if we have a transcription factor whose footprint occurs, whose motif occurs within the promoter regions of two different genes, we can be more sure that that transcription factor actually binds there if we see similar expression patterns between those two genes. Similarly, if we see in the promoter region of a single gene two transcription factor binding motifs adjacent to each other in the promoter region of that gene, we can be more sure that those two transcription factors cooperatively bind if there is data showing an interaction between these two proteins in the PPI data. So using these two concepts, John Quackenbush's lab, particularly Kimby Glass, developed PANDA, passing attributes between networks for data assimilation, which is a method for constructing a gene regulatory network, integrating these three different data types: a motif transcription factor to gene network, illustrating the presence or absence of transcription factor motifs in the promoter regions of genes, and then combining that with protein-protein interaction data and gene co-expression data from the particular tissue or cellular context of interest, and overlapping and combining these networks in an iterative fashion to arrive at a final gene regulatory network. And once these networks are constructed, you can construct these networks per individual using an interpolation kind of method called LINES, or you could construct networks per tissue or for healthy and disease. And then we want to be able to make comparisons between these networks to identify which genes are differentially regulated between the two conditions that we're studying. And the kind of metric that we use for this, we call differential targeting, which is interpreted as the extent to which a gene is differentially targeted by transcription factors in two different states. So for example, if we had a healthy network and a disease network, we would sum the inbound edges to a gene in the healthy network to arrive at a healthy gene targeting score, and sum the inbound edges in the disease network to arrive at a disease gene targeting score. And one of our recent applications of this type of investigation was in pancreatic cancer. This is a very heterogeneous type of tumor, and there are two main subtypes, basal-like and classical. And these subtypes are important for us to try to understand because it has been found in several different publications, including the original publication that determined these subtypes, that patients with basal-like tumors have significantly less survival, 5-year survival rates, and they also have differing response to treatments. So it's important to be able to understand what is different about these basal-like tumors, what makes them harder to treat, what are the molecular differences between basal-like and classical tumors, so that we can try to understand what should we perhaps try to target for treatment. So as an investigation, try to understand better what are the differential signals of gene regulation between basal-like and classical tumors. We used the publicly available resource of the Cancer Genome Atlas, TCGA, where there are 150 pancreatic ductal adenocarcinoma, or PDAC, samples, which have RNA-seq data. And we used the PANDA Linus sample-specific gene regulatory network approach to construct a gene regulatory network for each of these 150 patient samples. And then after dividing the tumors into basal-like and classical tumors, we calculated differential targeting metrics. So looked at the gene targeting score for each gene in each network and compared the differential targeting metric. We compared the targeting metric for each gene in basal-like and classical tumors and ranked these genes based on how different their gene regulation signatures are across basal-like and classical. And to get an idea of the functions which were showing up in these genes which were very differentially regulated, we performed a gene ontology functional enrichment and saw some very, very interesting functions coming up. Particularly, we found a lot of functions that we only see as being differentially regulated when we consider differential targeting as our metric of choice, which we do not see when we only look at differential gene expression. And examples of these functions that differential targeting has shown us is things like mitotic cell cycle, cell division, and then also cellular adhesion and migration related functions. So this is pointing us to some interesting biological pathways, telling us that perhaps these basal-like tumors are more aggressive because they have some characteristics about their growth and their cell cycle, and perhaps also their ability to adhere and invade surrounding tissues. So this is an interesting example in pancreatic cancer, and also a good illustration of how using these network-based regulatory metrics like differential gene targeting, as opposed to simply gene expression, can help uncover some interesting functional insight into differences between different subtypes of a disease, or between a disease and a healthy state. So moving on to the second part, we wanted to try to extend this approach to not only take into account transcription factor motifs, protein-protein interaction data, and gene expression data, but we wanted to try to allow the individual's genotype to be incorporated into the construction of their gene regulatory network, to tailor a network to a given patient so that we might see what impact the genetic variants they harbor can have on their own gene regulatory network. And so we want to use genetic variation that we may find in transcription factor motifs to generate individual-specific gene regulatory networks. And this is a method we published recently, which we call EGRET. And what we're trying to find here are instances where in one individual, a transcription factor would usually bind the promoter region of a given gene, and regulate the expression of that gene. Whereas in another individual, because of a mutation in that transcription factor binding site, this transcription factor is no longer able to bind the promoter region of the gene and is thus dysregulated. So looking at our existing framework here, we are looking to integrate individual specific gene or individual specific mutation data into these portions of this schema to be able to integrate these genetic variants into the transcription factor to gene regulatory network, which will then allow us to construct these individual specific networks to not only place the genetic variants of an individual within a regulatory context, but it will also allow us to investigate regulatory network properties which correlate with disease and potentially identify disease mechanisms, since the underlying construction of these networks is built around a particular mechanism of disrupted transcription factor binding. So to accomplish this, we used various resources. Firstly, we used cis-EQTL data from GTEx, which consists of genetic variants in the promoter, or at least near to genes, whose mutational status is correlated with the expression of the adjacent gene. And we also used QBIC, which is a prediction method which came out of Raluca Gordon's group at Duke University. And this is a method which is trained on in vitro protein binding microarray data to predict the effect of a single point mutation on transcription factor binding in vitro. And combining this all together, we call this EGRET, estimating the genetic regulatory effect on TFs. And EGRET takes various different inputs. So on a population level input, these are inputs that one can get from a public resource like the GTEx database or TCGA, because these are population level. They are not necessarily specific to the individual that you are studying, but they are the broad resources that we're going to interpolate from. We require gene expression data, transcription factor, protein-protein interaction data, DNA binding motifs of transcription factors, and EQTLs, expression quantitative trait loci. The only individual specific data that we require for this method is genetic variants, or the genome of an individual. And we developed this method using 2 cell lines. There was not a lot of data available that matched our criteria, but there were 2 cell lines which are derived from blood, which have both very high quality genomes and genotypes, as well as a wealth of ChIP-seq data available for validation of transcription factor binding. So the first step of EGRET involves constructing the EGRET prior. So this step looks for genetic variants in an individual which exist within a transcription factor motif in the promoter region of a gene, and which are also themselves EQTLs associated with the expression of the adjacent gene. And then we combine this with cubic predictions of that motif and that variant to see if in vitro, at least, there is a prediction that this change of sequence, this particular mutation, would affect the binding of that particular transcription factor at that location. And we quantitatively construct the network using edge weights. So our EGRET edge weights are constructed using this formula here. So our EGRET prior edge weight is the baseline edge weight based on the presence or absence of a motif in the promoter region of a gene. And then we subtract this absolute product of the cubic effect of a SNP, the alternate allele dosage, and the EQTL Baylor value. And then secondly, this prior is passed through the message passing approach of PANDA, combining this EGRET prior with both co-expression and PPI data to arrive at our final pruned and refined gene regulatory network per individual. So as part of this, we constructed 3 networks. We constructed a baseline network as almost a control group, which included no genotype information. And then we constructed a gene regulatory network for each of our 2 individual cell lines. We validated the edges in these networks using multiple public data sources, including ChIP-seq, allele-specific expression data from lymphoblastoid cell lines, as well as chromatin accessibility QTL data. And we quantified a slightly different variation of the gene regulatory differential targeting scores, which we call disruption scores, because they are quantifying the extent to which a gene's transcription factor binding sites within its promoter are disrupted by genetic variants. So the edge disruption score is simply the difference between the edge weight of the EGRET network and the baseline network. And the transcription factor disruption score is then summed per gene, per outbound gene from a transcription factor, to get an indication of the extent to which a transcription factor's genome-wide binding sites are disrupted by genetic variants. And the gene disruption score gives us an indication of how much the promoter region of a gene is disrupting the binding of all the transcription factors that would usually bind the promoter region of that gene. So in our validation, in the comparison of 2 genetically distinct cell lines, we found that high disruption scores were indeed enriched for differential transcription factor binding, as validated by ChIP-seq. They were also enriched for chromatin accessibility QTL variants, as well as variants which cause allele-specific expression. This is an example of one of the sites where we found differential transcription factor binding. So the purple track shows our eQTL that is within the RALA transcription factor motif in the promoter region of this gene. And the 2 orange tracks show the genotypes of the 2 cell lines. And we can see that GM12878 in the teal track, the RALA transcription factor binds in GM12878, but does not bind in K562. And similarly, we can see at the same position as that disruptive eQTL, we can see that that disruptive variant exists in K562 and not in GM12878. So this is an example of one of the disrupted binding sites that we found with this method and validated in the same cell lines through the public ChIP-seq data. And as an application of this method to a larger population, we applied the EGRET method to 119 individuals, the Yoruba individuals, for which there had been 3 sets of different cell lines developed, which are, at least for now, proxies for different tissues. So we have lymphoblastoid cell lines, we have cardiomyocytes, and we have induced pluripotent stem cell lines. And we have 119 individual cell lines of each of these different types, 1 per individual per cell type. So we constructed an EGRET network for each of these individuals in each cell type, as well as a baseline network, and then calculated our transcription factor disruption scores. And for each of these, we started to look for enrichment in potential disease genes that we could find in the GWAS catalog. In particular, we looked at Crohn's disease and coronary artery disease. And we found that transcription factor disruption scores were higher for disease-related transcription factors in the most relevant cell type. So for example, in cardiomyocytes, these transcription factors that had the highest transcription factor disruption scores, those transcription factors whose binding sites were disrupted by genetic variants, we found that those were dominated by, in signal, by a single transcription factor, ERG. And this is a member of the erythroblast transformation-specific gene family. It's involved in angiogenesis. And the high TF disruption scores for CAD genes, coronary artery disease genes in cardiomyocytes, were all driven by the presence or absence of a single variant on chromosome 1. And ERG, the transcription factor, if we were to look at the gene that it is regulating, the gene that it is binding the promoter region of, or that the binding of is being disrupted near to, is CSRP1. And this is a smooth muscle marker. If we look at its expression in GTEx, it is very high in all of our artery tissues. And looking in the literature of this gene, we see that it's associated with a lot of functions which we would associate with potential coronary artery disease, including the bundling of actin filaments, cardiovascular development, response to arterial injury. There was also an interesting study in zebrafish where knockdown of CSRP1 caused cardiac bifida. And also a frame shift mutation of CSRP1 has been linked to congenital cardiac defects in a large human pedigree. So our EGRET analysis has now supported a previously unreported mechanism for the action of ERG in heart disease, in that it regulates the expression of CSRP1. And our networks hypothesize that this regulation can be disrupted by genetic variation. So in summary, we've talked about how gene regulatory networks and using complex network measures such as the gene targeting metric or the disruption score can be used to uncover differences which we would not have seen before by looking at measures purely like differential expression. And I talked about the EGRET method, which we've recently developed to extend the use of gene regulatory networks, tailoring them to be individual specific based on the underlying genotype and the underlying mutations that an individual harbors. And then our proof of concept analysis of applying EGRET to a population of individuals, where we could see differences in individual specific regulatory networks, and that we found the tissue specificity of disruptions to be clear and seem to match well based on the tissue type we were looking at. And we're hoping to use this method further to continue to look at the differences between other cancers and healthy tissues, as well as particular subtypes of cancers. And I'd like to thank all my colleagues at the Lineberger Institute Comprehensive Cancer Center, as well as my old colleagues at the Harvard School of Public Health, particularly John Quackenbush at HSPH and Jen Ye at the Lineberger. So thank you very much for listening.